origin of life seemed virtually solved by a single simple experiment. Stanley L. Miller, then a 23-year-old graduate student at the University of Chicago, recreated the primeval Earth in a sealed glass apparatus. He filled it with a few liters of methane, ammonia and hydrogen, the atmosphere, and some water, the oceans. A spark discharge device zapped the gases with simulated lightning, while a heating coil kept the water bubbling. Within a few days, the water and glass were stained with a reddish goo. On analyzing the substance, Miller found to his delight that it was rich in amino acids. These organic compounds link up to form proteins, the basic stuff of life. Miller's results, which he published in a modest 2-page article in Science, seemed to provide stunning evidence that life could arise out of simple chemical reactions in the primordial soup. Pundits speculated that scientists, like Mary Shelley's Dr. Frankenstein, would shortly conjure up living organisms in their laboratories and thereby demonstrate in detail how Genesis unfolded. It hasn't worked out that way. The problem of the origin of life has turned out to be much more difficult than I and most other people envisioned, said Miller, then a professor of chemistry at the University of California at San Diego. Of course, there has been progress since 1953. That same year, in fact, James D. Watson and Francis H.C. Crick deciphered the structure of deoxyribonucleic acid, DNA, whose double-strand helix carries the information that cells need to build and organize proteins. Over the next few decades, experiments similar to Miller's demonstrated how the components of DNA, which are called nucleotides, as well as those of proteins, could have been synthesized under prebiotic conditions. These organic compounds could have accumulated in various bodies of water, in some warm little pond, as Charles Darwin once speculated in a letter, or in tidal pools or shallow seas. Experiments in the early 1980s seemed to complete the picture. They revealed that ribonucleic acid, or RNA, a single-strand molecule that serves as DNA's helpmate in manufacturing proteins, might have the ability to make copies of itself without the assistance of enzymes. Some investigators concluded that the first organisms consisted of RNA, and that an early RNA world had provided a bridge from simple chemistry to prototypes of the complex DNA-based cells found in modern organisms. According to the fossil record, such cells emerged within the first billion years after the Earth had formed 4.5 billion years ago. Although this scenario is already ensconced in textbooks, it has been seriously challenged of late. Tests of the RNA world hypothesis have shown that RNA is difficult to synthesize in the conditions that probably prevailed when life originated, and that the molecule cannot easily generate copies of itself. To make matters worse, recent findings suggest that life arose in an environment far less hospitable than Miller's glass apparatus. The primordial atmosphere may not have contained methane and ammonia, as Miller had assumed, and so it may not have been nearly as favorable for the synthesis of organic compounds as his experiment suggested. Moreover, studies of craters on the Moon indicate that the Earth was being repeatedly laid waste by huge meteorites and comets. It looks like life began not in a warm little pond, but in a raging tempest, says Christopher P. McKay, a space scientist at the NASA Ames Research Center in California. As the old paradigm has wobbled, many other theories, some brand new and some recycled, have been put forward. Observations of organic compounds in meteorites and comets have led to speculation that the raw materials for life fell to the Earth from outer space. The most extreme versions of this idea posit that not only inanimate organic compounds but also full-fledged organisms arrived from space. Some researchers, while holding that life